Hello. Today we're going to be talking about mutations a little bit, or I'm going to be talking about mutations a little bit, and then I'll have you do an activity and take a little quiz. Most of us have heard of mutations, have some idea of what it's about, but at the end of this, I'd like us to be able to identify different types of mutations, specifically in sequences of DNA, but realize that mutations can occur in any nucleic acids, and that includes RNA, but we're going to mostly focus on DNA. So we're going to start here with a little exercise, a little analogy that I like to use. If you read this here, you can see that we have a sentence. And you can see that the sentence is made up of 1, 2, 3, 4, 5, 6, 7 words. But of course, they're all kind of jammed together. But you might notice that the words are all 3 letters long, so hopefully that gets us thinking about, okay, why did Mr. J make these all 3-letter long words, and why are they all jammed together? Well, the idea here is to mimic DNA. And if you read this sentence now, right, the boy saw the dog run far, it makes sense. Even though it's kind of jammed together, we can still read it, and it makes sense, right? We read each of the words here. Right, the boy saw the dog, right? We see that, hopefully. Everybody can see that. The boy saw the dog run far. Okay. Now, imagine if we were to make some changes to the letters. Okay. So we're going to model what this would be like if changes had occurred to DNA. So for instance, let's eliminate the E here, and in its place we're going to put an X. Okay. Now, does that fundamentally change the meaning of the sentence? This word doesn't quite make sense, but the rest of the words, the other 6 words, still do, right? So even though the doesn't quite make sense, we can get the gist of it. But boy, saw, the, right? The rest of it's there. So that would be like a replacement or a substitution in DNA. So imagine we're looking at this, or if these were letters of DNA, and we replace one letter, it might not have that big of an impact on the meaning of that piece of DNA. Now, on the other hand, what if we were to eliminate, right, a letter altogether, right? A deletion. Now, when we try to read this in 3-letter chunks like we would codons, right, of an mRNA, it doesn't make sense anymore, right? THB doesn't mean anything. OYS, AWT, right? None of these make sense anymore in how they would be read, right? So you can see the whole frame has shifted, right? This is called a frame shift mutation. So now when we try to read it in letters of 3, it doesn't work anymore. Okay, one last example. Let's go back here and put our E back in, but let's now insert a letter. I don't know, let's just throw a V in there. Again, maybe certain aspects of it will still make sense. So here's the, but then we switch to the next frame. VBO doesn't make any sense. YSA doesn't make any sense. WTH, right? On down the line. So this is a good sort of analogy of how mutations can impact the meaning of DNA. When you change letters in DNA, that impacts RNA. And when the RNA is sent out to be translated, it doesn't make sense anymore, the amino acids that are put together because of that. So hopefully that wasn't too confusing and maybe helped you understand the idea of what mutations do to DNA. Okay, so a little bit more specifically, what are mutations? They're heritable changes in genetic information. So when DNA changes, can it be passed down to the next generation? Mistakes get made all the time, every single day. In fact, you're making, millions of mistakes every day are being made in the copying of your DNA. But it's usually fixed, right? There's enzymes that fix it. There are different types of mutations, right? Gene mutations would be the types that affect a single gene, right? So a change in the genetic material that affects a single gene would affect potentially a single trait. Chromosomal mutations, when you start changing a whole chromosome, you can imagine that's going to usually have bigger impacts, right? When a chromosome is changed. Okay, so what I just demonstrated in that sentence were point mutations. So this would be a change in one nucleotide at a known single point in a DNA sequence, right? This usually is going to occur during replication. So, right, when a letter gets substituted, usually that's not as big a deal. Look, see how it translates down here. We still end up with 3 out of 4 of the same amino acids. But when we do an insertion or a deletion, look what happens. The frame shifts, right? So every amino acid after that is not the correct amino acid. Okay, so again, just to kind of put it here in some other terms, substitution mutations are when there's a single change in the nucleic acid, right? So instead of a T here, or a C here rather, it gets replaced with a T. And then when you go to translate CAU, and it ends up giving you a different amino acid. But one different amino acid out of potentially hundreds of amino acids in a protein might not have any impact on the functionality of that protein. So that's called a silent mutation. It's not hurting anything, it's not necessarily helping anything, but usually it's a silent mutation. However, right, insertions and deletions, as I indicated, are what we call frame shift mutations. So now when you read in chunks of 3, like we know we do for codons, when you add a letter or take a letter out, that changes the 3 letters that are read, and that impacts the amino acids from that point on. This usually has a more dramatic impact on how well the protein functions after that if you change a bunch of amino acids in that protein. Okay, so again, many mutations have little to no effect on a person. They're happening in your cells all the time for all kinds of reasons. And we'll get into a little bit what some things are, what are some things that can cause mutations. But they can produce proteins with new or altered functions, which can be useful. So in fact, a lot of proteins have no effect or may even be useful. Okay, just quickly, just so you realize, right, there are chromosomal mutations too. And so this is really when whole sections of chromosome get changed, right? So if this little thing here represents a chromatid or an original chromosome, right, if you delete a whole section of that chromosome, which happens all the time, you can see these in karyotypes, then you usually lose several genes. And of course, if you lose genes, you lose the proteins that those genes made. Maybe it's an enzyme that helps clean up your lungs. Maybe it's insulin receptor. Maybe there's all kinds of things, right, that could be coded for in that language that get lost. Sometimes you get switches in information. Sometimes you get a translocation, material moves around. We're not going to worry too much about chromosomal mutations. I just want you to be aware that that changes can happen in chromosomes as well, not just in nucleic acids, not just in genes. Okay, so there are some harmful effects, right, of course, of mutations. One of the classic examples is called sickle cell disease, and that's caused by a single point mutation, a single change in a letter in the nucleic acids that code for hemoglobin, which is the molecule, the protein that carries oxygen for you. If you change it, it changes how that protein folds. So hemoglobin, instead of taking on a nice round shape, it takes on sort of a sickle shape, and that impacts how well it moves through your bloodstream. So this is an example of where a single point mutation can actually have pretty negative impacts on your blood's ability, your hemoglobin, the protein that carries your oxygen, its ability to do its job. Okay. Some cancers are also products of mutations. When we get a change in the uh, things that control how well a cell stops or continues to grow, remember the growth phases and interphase, there's proteins that control that. And if some of those proteins get mutated, get changed, that can speed up or slow down the growth of a cell. If you speed it up dramatically, that's generally when cancers can start. So mutations can lead to um, increases in cancer. And then finally, there are some beneficial effects to mutations. Some mutations have allowed organisms to grow more robust. Strawberries, for instance, are now polyploid. Um, Breeders sometimes look to sort of push mutations into plants to look for new traits. And so mutations are not always a bad thing. In fact, they can lead to good things. All right, I'm going to have you watch a short video after this and answer a few questions embedded in the video, and that'll be all for today on mutations.